And at this time, I'd like to welcome up Amy Perrin-Ross. If she could come forward. Amy is on our she's the chair of our clinical advisory committee. And she is also coordinator of the MS program at Loyola Medical Center. And she is also a 2012 inductee to the Health Professionals Hall of Fame. So congratulations to you. And then Amy is going to introduce our keynote speaker.
Thanks, John. It's my pleasure to be here today and to welcome each of you. I'm so pleased to see that we have yet again another full house. I'm really looking forward to the rest of today's program. In just a few moments, I'm going to be introducing our keynote speaker, Dr. Stephen Goldman, who will address progenitor cell-based treatment and modeling of myelin disease. Now, immediately following his presentation, there will be an opportunity to ask questions, not only of Dr. Goldman, but of a panel of local MS specialists as well. So, in order to do that, what we are asking is that you utilize the cards that are available to you to write down your questions, because we will only be taking written questions as opposed to trying to pass a microphone in the audience. The members of the panel's bios are listed in your program book, and the members of the panel include Dr. Jacqueline Bernard, Dr. Matthew McCoy, Dr. Susan Rubin, and Dr. Daniel Wynn. So again, all of these questions must be written down on the index cards, and they will be placed at your tables, as well as we will have volunteers who will come around and collect them at the end of Dr. Goldman's presentation. And I see Dr. Hintati in the audience as well. He's kind enough to do one of our breakout sessions a little later today, so we're glad that he's able to join us for our panel discussion as well. So now, without further ado, I'm really pleased to introduce our keynote speaker today, Dr. Stephen Goldman. Dr. Goldman is the Distinguished Professor of Neuroscience and Neurology at the University of Rochester Medical Center in Rochester, New York. He's the chief of the department's Division of Cell and Gene Therapy, as well as co-director of Rochester's Center for Transitional Neuromedicine. Dr. Goldman is also emeritus chairman of the Department of Neurology and holds additional appointments as professor of neurosurgery and as the Dean Zutz Chair in Biology of the Aging Brain. Dr. Goldman received his PhD from Rockefeller University and his MD from Cornell. So at this point, please join me in welcoming Dr. Stephen Goldman.
Well, thanks so much. Thanks for the wonderful introduction, and also really for the invitation to be here and speak. I work with lots of different advocacy groups and disease-oriented foundations, but I must say that video was a reminder there's just nothing like the MS Society. I've worked with the Society for the better part of the last 20 years, and it really is unique in American medicine in terms of the benefits that it has provided to the MS community. We've seen an explosion in not just MS research, but in research that has been brought to practice, an explosion in new technologies and treatments and medications over the last decade, as most of you here are aware. And that's in no small part due to the role of the MS Society. It has not only funded important work, but it has leveraged its activities in making sure that other funding agencies and support organizations have lent essentially their contributions to the fight against MS, and it's been just an extraordinary catalyst at the national and international level in terms of driving MS research. And I think we're all the beneficiaries of that. So it was also nice to see, as I was speaking with a number of the local neurologists yesterday and other folks here, that Illinois is such a wonderful really fertile ground for research, and you're really in an extraordinarily supportive population. I was hearing about the checkoff on the taxes for MS research. It's fair to say that would not fly in New York. It's really quite unique, and I think it's a benefit to all of you to be in such an open-minded and research-oriented community. So in any event, what I'm going to speak to you about today is a strategy we've been working on for the last 20 years in terms of regenerating certain cell types of the brain and adding certain cell types to the brain as a means of brain repair, as a strategy for repairing brain lesions. Okay, so in this first slide, many of you are at least familiar with what's going on here. I'll walk you through the picture. This is what an acute lesion looks like in multiple sclerosis at the microscopic level. A couple of things are going on here. So these are axons. Of course, the nerve fibers that are going from one area of the brain or spinal cord, as the case may be, to another. And these axons, of course, are the medium through which information is transferred in the brain and in the central nervous system more broadly. Multiple sclerosis results from, as most of you know, the loss of insulation around those axons. That insulation, which essentially allows an impulse to be conducted along a fiber, is provided by myelin, and that myelin, which of course we can think of as the insulation around a copper wire, well, it's the same concept. That myelin is supplied by cells called oligodendrocytes. And so here in this lesion, we have axons, but here and there, these axons they're surrounded by myelin in red, and here and there, those axons are broken. There's axonal transection that's occurring in the setting of the acute inflammation, the inflammatory process of MS. And you can see lots of spots where the myelin is degraded, some where the axon itself is lost as well, and then in some places what are called axonal spheroids. These are and you can see lots of them over here. These are basically the broken tips, and this is what's going to happen over here in the course of a week or so, when essentially the contents of the axon spill out and the axon membrane reanneals around it, and you get these spheroids, which are essentially are the mark of axons that are no longer functional. They've been cut, they've been transected. And so with this type of process occurring, of course, transmission is lost along the course of an axon, and then one part of the brain or spinal cord is no longer able to speak to another part, hence the great variety of symptoms and signs that we see in multiple sclerosis. So MS, of course, starts by, at least in the vast majority of cases, starts by occasional episodes of inflammation that become typically more frequent. Very often, and we've really come to realize this over the last decade or so as imaging technologies have become better and better, very often patients are having asymptomatic episodes of multiple sclerosis, or at least episodes are so minor that they don't really make much of it, symptoms that may come and go. And those represent small areas of inflammation and small areas of demyelination that are too minor, essentially, to be reported, as it were, in neurologic function. But finally, folks will start to have, as with more lesions accruing and larger lesions, they'll start to develop signs. And over time, they'll develop progressive impairment, progressive loss of neurologic function. Now, after the first couple of episodes of MS in the relapsing remitting phase, patients will get better. But what, as many of you are well aware, with each episode, patients will often and generally become less able to fully recover. So that there's some underlying damage that's occurring with each of these relapses, and that starts to accumulate. So much of that damage is due to axonal transection. So in some patients, axons have been lost. But much of it is also due to the chronicity of the demyelination. The myelin has been lost, and after a certain point, myelin doesn't grow back. It looks like, as far as we can tell at this point, a given region of brain can remyelinate a couple of times, but that's it. And that's because of the cells that I'm going to be describing that are in the brain that mediate that process of remyelination. They can do it a couple of times, but they can't keep doing it. And that's really the crux of this strategy for putting new progenitor cells into the brain, which we'll get to. Now, what happens over time is that disability increases. And as it does so, patients, of course, are losing more and more myelin. And at a certain point, myelin loss, it's not that it no longer is the problem, but it is only one feature of the problem, because at that point, patients will shift over from the relapsing remitting phase of MS to a period of more chronic neurodegeneration, where without the stepwise pattern of relapses and remissions, patients will start to undergo slow deterioration. And that's chronic progressive MS, of course, and just looking around the room, I could see that quite a number of you are suffering that condition. And this has been really our greatest challenge over the last decade in particular, because there have been so many new treatments that have been offered in terms of tackling the relapsing remitting phase of MS, the period of immune demyelination, that I think all in the field can now claim some really substantial success in gaining control of relapsing remitting MS. The problem is we have no control of this secondary process of neurodegeneration, which is very variable in individual patients in terms of when it starts. But once it starts, it really is what we call monotonic. It's slowly progressive, and we have little available to control it. A number of medications have been tried to arrest the progress of chronic progression in MS, and frankly, none have worked at this point. And so this is our challenge. How can we address this chronic neurodegenerative process of MS? And from that standpoint, we've actually started to think of MS as a neurodegenerative disease, much like the primary neurodegenerations of the brain otherwise. It's due to a couple of things. It's due to that axonal transection and axonal loss that has started to accumulate, and at the same time that results in progressive neurologic deterioration. But concurrently, that process of ongoing demyelination has robbed surviving neurons, robbed healthy neurons of the oligodendrocytes that they require and are dependent upon for trophic support. So axons, when they have a very close and intimate relationship with the oligodendrocytes, with the myelin that surrounds them. And so when an axon is denuded, when it's demyelinated, it's threatened, and it loses the nutritional and protective support that is supplied by myelin, and over a period of typically years, those axons that have survived the period of relapsing remitting MS go ahead and die. And so we're dealing with otherwise healthy axons that have been demyelinated that over time will die by virtue of not having oligodendrocytes to support them. And that's the fundamental issue that causes chronic progressive MS, and really the one that we're trying to tackle. And so this is basically the means by which we're trying to do so. And it gets back to work that I and others have been doing for a couple of decades now, looking at endogenous stem cells in the brain. And this work, and I just have to mention as a plug for basic research, starts when I was a graduate student. And I as a graduate student, somehow just wasn't excited about what I was hearing in terms of very classically medically oriented projects as I was shopping around labs. And then I found a lab, a very small one, that was working on the neurology of birdsong. Hey, I like birds. And so I thought, well, this is interesting. And so that's actually what my doctorate is in. Theoretically, it's in molecular genetics. Really, it's in birdsong, because we started to study how birds sing, and very specifically what the neurologic substrates for song were. And why did I find that interesting? Because when a male bird learns a new song every year, songbirds only learn songs in the spring, and they forget them in the fall after they've served their purpose of attracting a female. Well, when the bird's learning a new song, the part of the brain that controls song increases in size, and it lays down new cells. And that was really unique at that point. It was the only instance of structural neuroplasticity that we're aware of. And so I set about trying to understand what that process was about, and found that actually new neurons were being generated, and that was quite a surprise at that point. And so that got me and lots of other people interested in the potential for cellular plasticity in the adult brain. And of course, if neurons were being generated, they must be coming from someplace, and it turns out that they were coming from resident stem cells in the brain that persisted after development and were capable of generating new neurons. That set about a search by a number of groups in the 90s for analogous stem cells, neural stem cells in the brains of larger species of mammals, and but basically things marched up from mice to rats to monkeys to humans through the course of the 90s, as we and other groups found that, in fact, there were resident neural stem cells that persist in all vertebrate brains. And so what's a stem cell? We define a stem cell generally as a cell that can give rise to all the cell types of an organ. If it's a somatic stem cell, the type that I'm talking about here, a neural stem cell from the brain, means that that cell can give rise to all the cell types of the brain. And it can self-renew, meaning it can keep making copies of itself. Okay? So it can just infinitely go on making new copies of itself. At the same time, some of its daughter cells go ahead to become all of the cell types of an organ. When you're talking about, as so often is in the press, embryonic stem cells, that's essentially the same idea, but those are cells that are taken from very early embryos where the cells can give rise to all the cell types of the body, all cell types, and of course are self-generating. But those embryonic stem cells during development start to segregate into the different organs, and then each organ has its own somatic stem cell. So what we're dealing with here are somatic stem cells, and since we're talking about the brain, we call them neural stem cells. So it turned out that these neural stem cells are present in us. They're present in everything that's been looked at, from lizards and reptiles now, through birds, through mammals, to primates and humans. So this schematic gives you just a really quick idea of what we know in a very, very general sense about neural stem cell natural history in the adult human brain. And so we have these cells. They line the ventricles of the brain. Of course, the ventricles are the fluid-filled cavities inside the brain. The lateral ventricles are essentially the vestiges of the early vesicles from which the brain forms in early development. So the progenitors, or I should say the stem cells from which the brain forms in early development, line the ventricular system. And so what turns out is that when the adult brain forms, those cells never really go away. And so there's a small population of neural stem cells that remains within the ventricular wall of adult humans. There's not a lot of these. We estimate only about 100,000 cells are left per adult human brain. But these cells can then in turn give rise to new neurons, and those neurons can go into a few regions of the brain. In the human, this is a very minor pathway. We don't really utilize these cells for making new neurons very well, except perhaps after stroke or injury. That's a controversial field. And certainly in the area of the brain called the hippocampus, which is the area that we are always forming memories with. It's the area that is involved in memory acquisition and the initiation of memory consolidation. Well, it turns out that new neurons are being added to the hippocampus in an ongoing fashion. And as we get older, that process is probably becoming less and less efficient, and that's one of the many reasons why none of us have better memories as we get older. But what is really the dominant pathway in humans, and apes and humans both, is here. These neural stem cells, by far, most of them are giving rise to this cell type here. We call it here WMPC, the white matter progenitor cell. That's only because we first discovered it in white matter. But basically, these are glial progenitor cells. And they had been known to exist in lower species, but no one had ever been able to identify them or isolate them from human brain. And that's something we were able to do about 12 years back. It just turned out that it was a technological issue, effectively. A number of the markers that had been used in lower species didn't really work in humans, and so we identified the sets of proteins that human glial progenitor cells make, and use that knowledge to identify and isolate the cells. And then once we did so, we realized that these were really abundant, because when we started to separate them from the brain, we realized that whereas we were dealing with trivial fractions of a percent of all cells, literally thousandths of a percent of the cells in the brain or stem cells, between 3 and 5%, that's a huge, huge number. We're talking about tens of billions of cells, all the cells in the brain are glial progenitor cells. It's a very abundant cell population. In the white matter, it's about 3%. In the gray matter, as it turns out, about 5%. So these cells are a really active, resident population of cells in our brains, and they give rise to two cell types: astrocytes, the major support cell of the brain, and oligodendrocytes, of course, the myelinating cell type that's lost in the demyelinating diseases and in MS. So, of course, astrocytes are important in MS too, and this is something we'll get back to. when we say multiple sclerosis, well, sclerosis refers to the sclerotic plaques that the pathologists long ago found in patients' brains with multiple sclerosis. And so the name multiple sclerosis really refers to that pathologic definition of lots of areas of scarring in the brain. Sclerosis means scarring. And so the scarring is from astrocytes. So astrocytes will form scars in the brain in the setting of the inflammatory process of MS. And so both of these cell types are intimately involved in MS. Oligodendrocytes are the cell types that are needed and are lost. Astrocytes are the cell types that then react to that loss to form the scars that can be problematic themselves. So we focused on this pathway here, on the glial progenitor cell and the oligodendrocytes that derived from it. And skipping over a lot of work, once we isolated the cells, we first transplanted them into different models of demyelination in adult animals and found that they were, in fact, capable of giving rise to new oligodendrocytes that would then go ahead and myelinate axons in some of these experimental models of adult demyelination. And that's something we'll get back to. But then we realized, okay, here's something that looks promising. But when we started this work, we were using adult brain, because that's what we were studying, was adult brain biology. And we were dealing with samples like this, which are surgical samples, reasonably small, and we were isolating the glial progenitor cells from them. And that was okay for a proof of principle, but it wasn't something we could really use or scale up clinically. And so we switched to fetal brain. So during fetal development, during the latter part of the second trimester, that's when most glial progenitor cells are being made in the brain. So that this gives you an example here. This is 2 inches from here to here. All of the growth in the brain that occurs from this point, which is about 22, 23 weeks of fetal development, all the growth that occurs in the brain from this point on until we're an adult, from 2 inches to the size of an adult brain, occurs because of glial progenitor expansion and then the differentiation of those cells into astrocytes and oligodendrocytes and the myelination by the oligodendrocytes. All the neurons have been generated really by about 14, 15 weeks in human development. So the neurons are all there already. So everything that occurs after that is because of glial progenitor growth. Well, the glial progenitors are really active at this phase because they're providing that cellular substrate. So at this stage in development, about 2/3 of the width of the forebrain is composed of essentially pure populations of glial progenitor cells. And so this is the source that we focused on. Now these are frankly tragic cases. These are typically premature rupture of the membranes in the latter part of second trimester. Some are from sonographic failures that don't involve the nervous system. But these are all normal brains and obtained in medically necessary abortions. We established a network in New York State for obtaining these, and as a result, we have a reasonably reproducible and reliable sample availability of these types of tissues. And we're going to get to essentially alternatives that we've developed over the last few years, but we'll get to that towards the end of the talk. But this is this is the material, and these are the types of cells that we've used to essentially do all of the work that I'm going to describe to you now. So using that type of tissue, we can identify and then isolate the glial progenitor cells. And this is an approach called fluorescence activated cell sorting. Basically, the hematologists have been using this for 30 years. It's a way of separating cells based on proteins or molecules that are expressed on the cell surface, or in this case, maybe in the cell itself. So we use different surface markers to isolate the population of glial progenitor cells that could specifically give rise to oligodendrocytes. And they can also give rise to astrocytes, but what they cannot do is give rise to neurons. And so what we're doing here, without going through the detail, is separating the population that gives rise to neurons, which is identified by this marker protein here, and the population that give rise to oligodendrocytes, which is reported by this, or identified by this marker. Now, that told us, and this is work that goes back to about 2004 at that point, that we were able to get glial progenitor cells out of human brain tissue and to do it in large numbers from samples that were reasonably available and samples that had lots of these cells. And so finally we were able to scale up to do large-scale experiments. And so I mentioned before that we'd already looked at a couple of demyelinated models of adult brain tissue where the cells had worked. And so we thought, okay, let's really push it. Now that we can get these cells in abundance, let's see how far we could take this approach. And so we went to some very challenging models, if you will. And the one that I'm going to show you is one called the shiver mouse. And shiver, well, you can see why they're called shivers. They look like they're shivering, but really they're shaking. It's a primary truncal tremor and ataxia. These animals are very imbalanced, and that's how they start out neurologically, and we're going to see in a second they get worse. The problem with shiver mice is they don't make myelin at all. For the neurologists amongst you, they have an early stop codon on the first exon of the myelin basic protein gene. So myelin basic protein is a gene that's required for myelin formation. So the wrapping of myelin around an axon, the onion skin appearance, which we'll get some looks at later, of myelin, well, so that occurs because of myelin basic protein that essentially sticks one membrane to another in the oligodendrocyte as it wraps around the axon. And that's a process called myelin compaction. So without myelin basic protein, myelin doesn't form at all. An oligodendrocyte will wrap around an axon once or twice and then call it a day because it cannot compact the membranes. And so these mice start out looking okay, but as they grow older, they get bigger. And as they get bigger, axons become longer. Well, now these are axons that don't have insulation. And in the same way that an electrical wire will conduct electricity across a very short distance if it's not insulated, if it's a longer distance, it won't be able to successfully conduct. Same thing in an axon. As that axon gets longer, if it's not myelinated, there's what we call conduction failure. An impulse can't go from A to B. And so as the animal gets bigger, as it gets older, it gets more and more symptomatic. This is what the axons look like, and you can see this is shiver failed myelin. So these are oligodendrocytes and actually oligodendrocytic membrane that's surrounding these otherwise normal axons, but you don't see the kind of onion skin appearance. But that's important because we're going to show how we can get it. This is what these animals look like at 18 weeks. Now they very reliably die at 20 weeks. And so here we're looking at two of these. You can see how impaired they are. They can barely walk at all, but basically they have a little bit of forelimb strength that they use to get around. Hind limbs their legs are essentially powerless. They are extremely imbalanced. They're severe tremor. They're ataxic. And actually what you don't see here is that they're usually seizing. They start to seize at about 16 weeks. They develop really refractory seizure disorders by 18 weeks and 19, and by 20 weeks they die. And so it's an extremely challenging model. They make no myelin whatsoever. They've have been looked at for 20-odd years. Nothing had ever been done to give a shiver mouse an extra day of life. But it had been a very useful model for seeing whether cells could generate myelin, because if there's any myelin that forms, you know that it's from the donor, from the cell that you put in. And so Martha Windrum in the lab did this experiment back in 2004. She took the cells, the glial progenitor cells taken from fetal brain, and injected them into baby shiver mice. Okay? And the idea was to see whether we could engraft the baby's brain with enough of the progenitors that they would basically compete effectively with this diseased, if you will, with the genetically deficient glial progenitor cells in the mice themselves. So here, one of these animals killed at 3 months, and we see all the red cells here are human. These are very low power sections through the brain, front to back. And all of the red stain here, these are actually, there's lots of little dots, and every one of those dots is a human cell. And these are basically outlining the white matter tracts of the mouse brain. So it told us that the cells really were migrating and effectively getting around these brains. Now, that was good news, but when we actually followed those animals, when they got to 20 weeks, they died. And in fact, skipping ahead a little bit, we did look at the brains, and we saw that myelin was being made. But it didn't seem to matter. The mice were still dying at 20 weeks. And so we went back to the drawing board, and it's led to a lot of interesting science in the years since to see what was going on. If we were getting myelin, then how come we were only getting myelin, as it turned out, in some parts of the brain, but not most parts? And the cells yet were getting all over the place. Well, it turned out, making a long story short, that when the cells migrate through the brain, they're seeing all sorts of different signals. And if they migrate through normal gray matter, they lose the ability to make oligodendrocytes and instead become astrocytes, okay, the scar-forming cells. And so here we needed to be able to get the cells delivered only into the white matter tracts. And the cells were going to migrate wherever they wanted. They would go into the gray matter as well and become astrocytes. But we weren't worried about that. What we were worried about was that when they made it through gray matter to get over to the next white matter tract over, they would have lost competence. They would have lost the ability to make an oligodendrocyte once they got to that new region. So we realized that we had to inject cells into each of the major white matter tracts. And so in order to have cells dispersed everywhere without relying upon contiguous spread, in order to get cells to where they needed to be. And so that was kind of a daunting task, or at least we looked at it initially that way and thought, well, gee whiz, how are we going to inject cells into all the major tracts? But it turned out that the cells do migrate everywhere, so that if you pick just a few very well-chosen spots in the nervous system and put the cells in, they'll migrate everywhere, and yet you can achieve complete spread throughout the major white matter tracts without any cells having to leave white matter to get to go through gray matter to get to the next white matter tract. Okay, so it's an interesting problem in geometry that it turned out that 5 sites were sufficient to inject in the mouse brain to achieve this purpose, and we've modeled it since in humans. 6 sites we expect will be sufficient in baby humans, and just 4 in adult humans. So that Martha Wyndham then went ahead and did this study, which was to take this 5 injection series and injected these baby mice again with the human cells, and then we looked again. And we looked far out. We looked at a number of different time points, but this just shows you what it looks like at 35 weeks. This is a little dark, so maybe a little hard to appreciate, but there are red cells everywhere in this brain. So this is a single, what we call sagittal, sideways section through the brain. And every one of the small red dots here is a human cell. This is just a single 14 micrometer thick section, so it's the thickness basically of a single cell, and yet you see how many cells have really engrafted the brain. So what appeared to be happening was the cells really were migrating everywhere. They were doing it nicely. And if we waited long enough, they were getting throughout the entire nervous system. And when we looked for myelin, we found plenty of it. So here we're looking at an adjacent section, and this is basically the normal myelin pattern of an adult mouse, except this is a shiverer mouse. It doesn't have any myelin, so we know that this is all donor-derived. It all came from the transplants. And we also, this kind of cuts to the chase, this picture was taken at 9 months, and I mentioned to you before, these animals all die at 20 weeks. Well, this one was at 35 weeks. And the myelin was normal. And that's really the bottom line. So looking at it ultrastructurally, it was normal myelin, but this is human myelin around the mouse axons. These are nodes of Ranvier, which the neurologists amongst you will be familiar with. These are the areas along an axon at which electrical conduction, basically saltatory conduction we call it, is occurring. And so as an oligodendrocyte wraps around an axon, it wraps over a given segment, and then there's a space, and then there's another segment. And it's at those spaces in between oligodendrocyte ensheathment segments, in those spaces, that's where the ion channels cluster that cause the electrical conduction to occur. In the shiverer mouse, as well as in any demyelinated region, and that includes in MS brain or spinal cord, when the oligodendrocyte is lost, those ion channels aren't what we call sequestered, and they spread out along the membrane. That's one of the things that makes the axon effectively lose function. And so regaining nodes of Ranvier is really a critical function, something we looked at as a real endpoint. So here we're looking at, without going through all the details, but the red stains here, that's the clustering of the sodium channel, the fast sodium channel that basically allows electrical current to pass through the axon. And so here we're seeing the clustering of that sodium channel, and that's telling us that the nodes of Ranvier are there again, that they've been reformed in these mice. And there are a number of proteins that we use to define normal nodal structure, and these are all normal. And that predicted that these axons should be working again, that they should be able to conduct an impulse more rapidly. And it turned out that was the case. So here we're looking, don't worry about the tracings here, but the bottom line is looking at how they aggregate. This is the conduction velocity across the corpus callosum, the major tract going from one side of the brain to the other. So it all comes down to how quick transmission occurs across that tract or any other. And so here in the normal mouse, we see that the impulse goes at just over 300 millimeters per second from one side of the brain to the other. In the shiver mouse, that's not so much the case. It's just about 200 millimeters per second, much, much slower. But in the transplanted animals, at this time point, no different than the control. And so that told us that, gee whiz, the transplants really looked to be sufficient to regenerate the nodes of Ranvier, to make normal myelin, and to restore normal conduction velocity to these axons. And so that predicted, now that we had this injection protocol that allowed the cells to go everywhere and remain competent to become oligodendrocytes, now that we had all those pieces in place, well, the animal should live longer, and we've already seen an example of one who did. So this was a large survival series. There was about 150 animals in this, 50 per group. And the white line and the green line show different controls. One that didn't get anything, just normal shivers, and the other received saline injections. And they all die like clockwork, pretty reliably, about 150, 160 days, a little over 21 weeks. But when we look at the transplanted animals, we see that about a third to a quarter are rescued. Okay? It's all or none. They get to a certain point, and then they survive. So anybody who gets to this point and can survive beyond 170 days or so, then lives. We had a few who we killed for histology. I mentioned yesterday that we found out that we were starting to think of these as super animals, basically. We found out they're not so super. If somebody drops the cage, they will die, and so we lost 2 here. But then the others we followed, and we killed 4 of them here, and then we let the others go, and they went out to 2 years, which is the normal lifespan of the animal. So this was a complete rescue. But we wanted to really understand why. Okay, this is good news, but what about these guys? Okay, so more than 2/3 of the animals still died. What was that about? And so we realized it was all about time. So here we're looking at the seizure incidence of these animals. I mentioned to you that they normally will die of refractory seizures, of status epilepticus. And so when we looked at and quantified the incidence of seizures in these animals, we saw that the transplanted and the untransplanted animals both seized, because the transplanted animals at this point still weren't benefiting from the transplants. But the animals that survived, the rescued animals, totally stopped seizing. So we realized that was key, and really focused on this region here to see what was going on at the 150, 160-day time point. And what turned out to be the case was that the animals who were surviving that period were myelinating more quickly, and very specifically some regions, for the neurologists amongst you, some regions in the ventral midbrain that looked to be now we realize some of the interlaminar nuclei are the seizure generators in these animals, and again, that's a point for the neurologists here. But it was interesting, and what that means is that speed was absolutely of the essence, and so this really trivial differences from one mouse to another. If these regions that basically controlled epilepsy myelinated quickly enough, those animals would live, and if they didn't myelinate quickly enough, the animals would die of their seizures first. And so that told us that speed mattered in a big way. And then, the flip side of that is, we're looking at events at 150, 160 days, 20, 21, 22 weeks. Why isn't this happening more quickly? Why aren't we myelinating from these cells more quickly? We're putting them in on the day of birth. So then we started to look to see how rapidly this process was occurring. And this really gives you a good sense of things. At 3 months, 5 months, 9 months. The red here is neurofilament, which these are the mouse axons in red, and the green shows the myelin. And so at 3 months, we could see the human oligodendrocytes in blue are there. They've been grafted nicely. They've been generated from the progenitors. They're making myelin. They're just not making a whole lot of it. Right? There's only about 10% of the axons are actually myelinated here. By the time you're at 5 months, about 50% of the axons are. This is about the point where the animals were dying. And so that told us that we had to have at least this proportion of axons myelinated. And this is in the forebrain. This is not the area of the brain that was really critical in terms of defining death, but this was a useful region for us to use to compare across brain regions. So we knew that we had to have at least this much myelin, but by the time we got out to 9 months, basically all the fibers were myelinated. And so we thought, it's good news and bad news. So we can myelinate all the fibers. It just takes 9 months to do it. So what we realized was happening, it seemed awfully slow, but it really wasn't. Oligodendrocytes in the brain basically will typically myelinate at least 50 and usually over 100 axons per oligodendrocyte. And so these are really busy cells. They will approach an axon, they'll wrap around it, 15, 20 times, and then go off to the next axon. Well, that process of wrapping around a given axon takes, as it turns out, a day and a half to 2 days. So it's not like the oligodendrocyte isn't busy. It's just that it takes a day or two to myelinate a given axon, and yet that oligodendrocyte may have to myelinate 100 axons plus. So, do the math, it's a couple hundred days for a given oligodendrocyte's domain to be fully myelinated. So it's a slow process, and we realized we had to be able to speed it up if this was going to be useful, and yet if we could speed it up, the animals who survived with absolutely fully myelinated normal nervous systems. This is one animal we killed at 13 months. That's all human myelin, and it's an entirely normal nervous system. Another animal basically looking at different regions of the brain, and here you get an idea of just how heavily engrafted these are with the human donor cells. The human cells turns out to be an advantage of this experimental system. The human cells take over; they outcompete the mouse cells, and which has all sorts of other implications that have been fascinating. We can discuss in the question and answer session. So these cells get throughout the entire nervous system, not just the brain. So the spinal cord, this is just one section of the spinal cord, posterior columns, longitudinal fibers, fully myelinated. This is all, it's a black and white, but this is all normal myelin. And even the roots, so the spinal roots, the cranial nerve roots, everything in the central nervous system myelinates. But the peripheral nervous system doesn't. And this has a lot of implications clinically not for MS, but for a lot of myelin disorders, genetic myelin disorders that affect PNS and CNS both. So here we're looking, just by way of example, the trigeminal ganglion. So it's one of the roots and the ganglion that comes off of the brainstem that controls sensation to the face. And so here, this is central nervous system, and all the red cells, the purple cells, are human, and we can see that they've gone right out to the border of the ganglion, which is central nervous system, and the nerve, the fifth nerve. And in the ganglion, they've myelinated, but they have not gone into the nerve at all, and they, of course, haven't myelinated the nerve. So there's an absolute border at the central nervous system and peripheral nervous system that the cells respect. We think it's fascinating. We don't understand why it happens, and that's something a number of the folks in the lab have been working on, is try to understand what those signals are. Bottom line is that cells get everywhere in the nervous system, and at least the central nervous system, and within CNS can fully remyelinate anything. And so what happens then, when we look at one of these animals, in this case at 13 months, now just picture the animals we saw before. This was one of them, so that at 18 weeks, this guy was quadriparetic, seizing, blind, and here we are, 13 months, he's been rescued, strength is back, he's walking around okay, vision's back. He still has a little bit of ataxia, he's still a little, this is not a mouse you're going to see running across your kitchen floor, he's still a little bit imbalanced. Or if he does run across your floor, you'll catch him easily, but otherwise he's a pretty happy mouse. And so this told us that there was really remarkably a functional recovery that was being made possible here as well. It wasn't just that we were seeing structural reconstitution, but literally the nervous system was regaining all of its lost functions, or not all, but many of its lost functions in that process. So we were really starting to focus on, again, this need for speed. Well, we can't really speed up the underlying biology without introducing other potential issues. The glial progenitor cells are interesting not only to folks interested in myelin diseases, they're of interest to neuro-oncologists as well, because these are the cell types that can give rise to gliomas, to primary brain tumors. And so we have all these glial progenitor cells in our brains, as I mentioned. Well, some of them go awry, and in the rare cases when they do, one can see tumor formation. And so when we start to manipulate these cells to try to speed up development on any level, one of the things we have to worry about is triggering one of these processes that's potentially tumorigenic. So we don't want to speed up development. We want to take advantage just of the normal natural history of the cells. So given that, how do we speed up the process of myelination if we can't speed up differentiation of the cells themselves? And so one option was to try to select out from the progenitor population those cells that were the most biased towards quickly becoming oligodendrocytes. So when we take tissue, when we take the cells out of fetal tissue, we're getting a whole range of stages, whole range of ages of individual cells, and we don't really know what we're putting in from that standpoint. Some of those cells may have months before they can become an oligodendrocyte and myelinate. Some of those cells may just have a week and they'll become an oligodendrocyte and start to myelinate. So we went through, and I'm going to skip over all the details, but we went through a set of experiments where we looked at the different genes that were being made by the glial progenitor cells as a population, and then focused on genes that we associated with oligodendrocyte maturation. And then we did what's called a cluster analysis, and we identified the genes that were associated, that clustered with those oligodendrocyte genes, and then predicted on that basis what proteins those genes were making and what proteins would be expressed on the surfaces of the cells that were most likely to become oligodendrocytes quickly. And then we developed sorting techniques for pulling those cells out. And so that was one long sentence. I apologize, it was a run-on sentence, but there was 7 years' work in that sentence. But the bottom line is that it worked, and so we ended up with the ability to identify and then isolate the glial progenitor cells that were already essentially primed and tuned to become oligodendrocytes. And when we transplanted those, and those were identified by a marker some of the neurologists here may be familiar with, called PDGF-alpha receptor. And there's one specific epitope on that receptor. It's called CD140A. And that's what we used to separate the cells. When we transplanted them, we found that they migrated just as well as the cells did, the cells that we discussed so far, which were separated on a marker called A2B5. Well, they migrated just as well, but they did it more quickly. And by just 12 weeks, here we're looking at a section right across the front of the brain of a mouse, a shiver mouse, and that's one side of the brain, that's the other. This is the corpus callosum, the major tract going from one side to the other. And here it's completely remyelinated at just 3 months. And so when we look at high power, we see in this case the colors. So the myelin is red here, and it's red here, and the axons are in green. Well, this here, E, represents a CD140 transplanted shiver mouse. So these more rapidly myelinating cells have been put in here, and we see at just 3 months, over half of the axons have myelinated. Remember the slide I showed you before, 3 months with that A2B5 injected pool, only 10% had. And yet here's one of those A2B5 injected pools here that we use as a control, and again, less than 10% of these axons are myelinated. So this was the point-to-point, the head-to-head comparison, and we realized that this population here was a much, much better cell population to be injecting. They still give rise to astrocytes as well as oligodendrocytes. It's just that they make that decision to finally commit one way or the other much more quickly. They only have a division or two left in them at that point, and the result is that you get these actually very pretty images, but this admixture of myelin, human myelin in red, in this case, human astrocytes in green. Now, these are fibrous astrocytes, which are the astrocytes that support oligodendrocytes, that support myelin. These are not the astrocytes that one typically sees in scars. And so you can look at that as these are the good astrocytes. They're necessary for oligodendrocyte function. So here we're seeing both of them co-generated and essentially able to reconstruct the entire white matter of these animals, and to do it very quickly in just 3 months. So at that point, we ran another survival study and found that, at least in a much smaller set of animals, that in fact these cells were capable of really substantially rescuing the population of animals into which they were injected. So that told us that this was a really nice cellular vector, we call it, a cell population for transplanting into demyelinating diseases. But demyelinating diseases of children, of neonates, of everything I've shown you so far has involved the injection of these cells into baby mice, right? And so we realized, okay, this is a great cellular vector, we think, but how's it going to behave in adult brain? Is it going to be able to migrate in the adult brain just as well as in baby brains? Is it going to be able to remyelinate demyelinated lesions, which are focal, or diffuse as the case may be, but not as widespread as in these models of pediatric myelin disorders. And so we injected the cells, this is that CD140, I'll keep calling it, cell population, into adult mice. And in this case, we put them into adult shiver mice, and we saw the cells actually migrated just as well as they did in baby brains, every bit as well, and they went ahead and myelinated just as well too. So this is an adult shiver, and this was actually really remarkable for us. So the green, of course, is myelin here. The red are the cells that here are, you can actually catch them in the process of generating new progenitors and sending progenitors out to the white matter to myelinate. And yet, this was really remarkable because these shivers normally would, as I mentioned before, would normally die at 20 weeks. They're rescued if they're injected neonatally. Here we've started to see animals do much better who were injected as adults after they were already symptomatic from their demyelination or from their hypermyelination. And at high power you can see that there are a lot of, I'm not sure what that is, but there it goes, good. Lots of red cells here. These are the human cells in red, and in this image the red cells are basically glia that are providing that substrate again, and myelin basic protein in green, and we can see the crossing fibers in the callosum. So this is very dense myelination in the adult animal. So we've also done this in chemically demyelinated models where we've taken the animals and given them toxins to destroy the myelin and then put the cells in, and they in fact will migrate and remyelinate those areas that have been demyelinated. I've used different variations in that theme. There are a variety of different toxin models. I'm not going to go through all those here, but just to give you one idea. So again, this is a little dark, but there are a lot of red cells here, and the red cells have been injected right here. And they were injected, these are all the human cells, in this case into an adult rat. We've done this with monkeys as well. In fact, essentially with the same data in the monkey as the rats, just we can run, for obvious reasons, many more rats than monkeys. So here we inject the cells here, and they can migrate throughout the white matter, also into some areas of the subcortical gray matter. In this case, this animal received a toxin injection over to this side of the brain a month before that. It's a toxin called lysolecithin that we and many other groups have worked with that results in the demyelination of areas in which it's placed. And so in this case, the cells have been injected. They've migrated throughout the white matter and some of the gray matter. They've also migrated into the area that had been previously demyelinated. And when we look at that area, histologically, we see that the myelin has been restored. So every red cell here, you can see the small red dots, at least if you're in the front of the room you can. Those are the human cells that have been grafted and basically replaced the demyelinated population, in which the mouse progenitors have largely been lost as well in the area of demyelination. So all the cells that have migrated in, the human cells that have migrated in, are essentially subserving the remyelination of this demyelinated lesion. Now some of this area up here, there's an admixture of human and endogenous mouse cells. These kinds of lesions will remyelinate on their own accord from the endogenous mouse glial progenitors that will migrate in from areas that have not been demyelinated. And so this is likely mixed in terms of the contributions of myelin from human and mouse up here. Down here, looking at it on a cell-by-cell basis, essentially all the oligodendrocytes are human. And so this tells us that this is a strategy that we can use just as well in adults as in pediatric and childhood myelin targets. And so we end up with this fairly broad list of disease targets. Lots of pediatric disorders of myelin. These are genetic hereditary leukodystrophies, we call them. Every one of them is uncommon. Some of them are quite rare, but as a group, they're not so uncommon. There are just lots of these individual enzymatic disorders and structural disorders of myelin. We look at all of them as potentially treatable with this strategy. Among other things, these cells that when we put them in are normal in terms of their enzymatic complement and function. So that some of these are enzymatic deficiencies that, if anything, the donor cells will not only regenerate myelin, but actually provide the enzymes that have been lost by virtue of the genetic deficiency. Cerebral palsy is an interesting case as well, because most cerebral palsy cases we realize now are in part a product of loss of oligodendrocyte progenitor cells. And so here we're thinking in terms of replacing these cells, and that may actually benefit CP as well. It's a much larger, obviously, disease target than the pediatric leukodystrophies, the hereditary disorders. But in adults, which is really the main topic for today, the vascular leukoencephalopathies, we call them, basically white matter stroke and white matter loss from small vessel disease and diabetics or hypertensives. These are all possible recipients of this type of progenitor transplant strategy, but really where we focused, of course, has been on MS and as we start out in chronic progressive MS. And so we've gotten to this point of setting up a clinical trial using glial progenitor cells, very specifically for remyelination in chronic progressive MS. And this is a trial, it's a consortium amongst all the upstate medical schools in New York, where we're going to take these CD140 sorted cells that I mentioned from fetal brain tissue and inject them into patients. These are intracerebral injections, so the same way that the mice and the rats and the monkeys have to get several injections into the brain through small burr holes, well, that would be the case for the humans as well. For patients. These are, it's not as bad as it sounds. These are basically very thin straws that are injected through small holes that are drilled, and cells are delivered directly into the targeted white matter. And with 4 injections planned per patient, all at once. This would be set up as what we call a phase 1-2 study, so that phase 1 in FDA parlance means a safety study, phase 2 dose determination and initial efficacy determination. Well, we're going to try to kill 2 birds with 1 stone and establish both the safety of the technique and get some basic dosing data. And it's not a large trial, just 20 patients initially, basically a 30-patient trial overall, 20 grafted and 10 controls, and we expect to start next year. And this is something that has been funded now, and we're putting together everything for the FDA at this point. It's not something where we're going to need a whole lot of cells. Basically, about 3 billion cells is enough for the whole trial, and yet that's a lot of cells if you're dealing with fetal tissue. So we started to ask, and I'm going to close with this thought in just a couple of slides to follow. But we started to ask, okay, if this works, we're actually pretty optimistic that it will. If this works, then how are we going to be able to scale this up to large patient populations when this whole trial only needs 3 billion cells? That's only about 3.5 grams or so, not a whole lot. And yet it's hard to get that from these samples. We'll actually need a number of samples of fetal tissue to be able to get this many cells. So how are we going to do that? And how are we going to be able to scale up to larger patient populations? And so what we plan to do is to generate these cells from embryonic stem cells and from a type of essentially a variety of pluripotential stem cell, I should say, called the induced pluripotential cell, which is a cell that, these are stem cells that are derived from any cell type of the body, where we then introduce what are called reprogramming genes to make those cells into the equivalent, if you will. They're not really equivalent, but into an analog of embryonic stem cells. And so we can take skin, we can take blood. This is a technique that was first worked out by a fellow named Shinya Yamanaka a couple of years back, but it's really revolutionized stem cell biology. We can take any cell type of the body at this point, but we're focusing really on easy-to-get cell types like skin fibroblasts or blood mononuclear cells, and by putting in a couple of what are called reprogramming factors, a couple of genes, essentially that drives development, if you will, in reverse, and we end up with cells that are stem cells that are capable of giving rise to any cell type. So we've worked out the methods for generating glial progenitor cells from those skin-derived and blood-derived stem cells. And so this is relatively recent work, but it's already convinced us that this will be the way to go in the future. So we can take skin cells, make them into iPS cells. These stem cells are called iPS cells, and then make glial progenitor cells from those iPS cells, and then make myelinating oligodendrocytes from those. Again, that's a long sentence, took a couple of years, but it's unfortunately it takes about 200 days to do for the entire technique. This is something that doesn't happen overnight, but at the end of the day, we can take a piece of a given individual's skin and give that person back his or her own oligodendrocytes in large numbers as well. When we inject them into shiver mice, we see myelination, we see astrocytic production both, just as if we're using tissue. These are what the individual myelinated oligodendrocytes look like. This is the myelin that they make. It's as absolutely prototypic as any myelin one will ever see from tissue or normal tissue, but this is all derived from skin. And when we go into shiver mice, we can completely remyelinate the nervous systems. We get survival curves that actually represent an almost complete rescue of the population. We're pulling off animals along the way for testing, and actually it turned out that there was a little more to the biology here, so that when we first did this, we took all cells out at different stages from the cultures, and then we realized that the survivors that were really rescued were surviving from transplants that were taken at a certain time point in vitro differentiation. And of course, since then, we've focused on that time point. So that's a strategy that looks to be even better than using tissue. We actually have better results and it's more controllable using these iPS cell-derived glioprogenitor cells. It gives us greater controllability, but over and above that, we can generate as many cells as are needed, literally in the hundreds of billions already. And we can do it in an autologous fashion, meaning on a patient-specific basis. We take a person's cells, generate all the oligodendrocytes or glioprogenitor cells that are needed for transplanting back to that person, so that at that point we don't have to necessarily worry about immunosuppression, because it's the person's own cells that will be delivered back. So we have a number of different ways to get glioprogenitor cells now, transplantable glioprogenitor cells from fetal tissue, from human embryonic stem cells, work that I didn't really talk about today, but form the prototype for methods for generating the cells from iPS cells. Again, these are somatic stem cells taken from skin or blood. There's a newer technique that's actually been described called direct induction, where you take the skin cell and make the glioprogenitor cell directly without going to a stem cell stage. That's actually very interesting from the standpoint of the cell biology. It's not a useful cellular vector for transplant purposes because one cannot expand the cells up and grow large numbers, but it is very interesting for some specific conditions. It may actually prove useful. But at the end of the day, we end up with these essentially preparations of glioprogenitor cells. We can purify the specific cell types of interest from them and then inject either into adult or embryonic or as the case may be, pediatric targets. But of course, our first clinical trial will be in a chronic progressive MS using these cells. And our very strong hope is that by the time we have initial data arguing for the efficacy of this approach, which could be really as short a time as really 3 years from now. Because the trial, we would, at least if everything stays on schedule and if the FDA gives us our blessing, we would hope to be able to start the trial in about 1.5 years, and then we would have the first efficacy data about 1 year after that. If things move as quickly as we hope, then we could be at the point, by the time we have that initial efficacy demonstration, of having iPS cells that are appropriate for the purpose as well, in which case we would quickly be able to scale up to larger populations. At least that's our hope and plan. And so this is the cast of characters, and it's only actually a very abbreviated list of the cast of characters who have been involved in this work over the years. And I just wanted to give you a quick idea here. The Upstate Consortium that I mentioned before, these are the folks involved in the clinical trial. These are all senior investigators at the 3 institutions that cover basically the clinical trialists, neuroradiologists, physicists, the epidemiologists, neuropsychologists, and genomicists, as well as the neurosurgeons. It's an extraordinarily involved procedure, as it turns out to be, in terms of doing this first-in-man type of strategy for essentially for cell replacement and cell repair in the brain. But we have every expectation that it'll be well worth it. I mean, to give you an idea, we have over 3 dozen senior investigators on the study that ultimately will transplant just 20 patients, and so we're running almost a 2-to-1 ratio for investigators to patients on this one. But I'd be very hopeful that as things pan out, and if we do see that this is as effective and promising as we hope, we'll be able to scale this up so that this will essentially be the core group from which we hope the strategy will very quickly, once we see efficacies, be able to spread. And amongst the different folks different organizations, I should say, now folks that have supported the work over the years, the MS Society really has been absolutely critical. They started to fund this work when all we were looking at were animal models of pediatric leukodystrophies with no reasonable expectation that it was going to be work that would be applicable for MS, and we were doing that based on essentially basic science that had been demonstrated only in canaries at that point. And so they, in a leap of faith, about 18 years ago, the MS Society started to fund this work and has been funding it ever since. And of course, there have been lots of other funding agencies that we've added on along the way. But it has always been remarkable to me how far-sighted and loyal the MS Society has been as an organization. It's that degree of foresight has been really critical to its support and advocacy for research, not just in my group, but in all the hundreds of groups that are funded in the U.S. that have really led to this explosion in not just research but clinical progress in multiple sclerosis. And I think I certainly owe a great debt of gratitude, as I think all of us here do, to the efforts of the MS Society. Thank you.